Hey microbiology students, we're starting a new lecture series this week and the topic is eukaryotic pathogens. So for the first mini weeks actually, so we are now in week 14 of the semester, and in all of the prior weeks we focus on bacteria. And of course, bacteria, as you know, are prokaryotes. And as I told you at the beginning of the semester, for microbiology class, bacteria are the star of the show. We do labs where we grow bacteria, we talk about their cell structures, and in the last unit, we talked about the diseases caused by certain pathogenic bacteria. Now, we are moving into a whole nother realm of microbiology. We are going to focus in the next 3 lectures on eukaryotic pathogens. This lecture is going to focus on pathogenic fungi. So recall, fungi are eukaryotic microbes. They are in their own kingdom in biology, only some of which cause disease. And actually, many fungi live inside of your body as a normal part of your microbiome, acting as mutualist and commensal fungi. So it's possible that these normal flora fungi can become opportunistic pathogens. We'll get to that on the next slide. We'll talk about reasons why they would become opportunistic. In this lecture, we're going to focus, however, on the fungi that do cause disease. And fungal diseases are referred to as mycoses. Some features of fungal mycoses, usually chronic, meaning that the disease lasts several weeks or longer, and it takes about that long to also recover from a fungal mycoses. This is because fungi, unlike bacteria, grow slower. Fungal reservoirs include both non-living, so environmental reservoirs like in soil and water, as well as inside of living organisms. Modes of transmission are usually indirect contact. So usually acquiring a pathogenic fungi from touching an object that's contaminated with spores from the fungus. So remember, that's fomite transmission. It's possible for spores to also be airborne, where we breathe in the spores and the spores germinate in the lung tissue. And then it's also possible for these opportunistic pathogens that reside inside of your body to take hold and become pathogenic. So those are the usual modes of transmission. It's also possible for pathogenic fungi to invade your digestive tract, so to eat food that's contaminated with spores, for example, and to have a gastrointestinal fungal infection. And it's also possible for the reproductive system to also be invaded by fungi. So now let's talk about those risk factors. So when would these normal fungi of the body become opportunistic pathogens? So we really consider these to be the top risk factors for that scenario. So infants and elderly, those with weakened immune systems or immunosuppressive health conditions like AIDS. People who have been on extended antibiotic use. This is because your microbiome consists of both bacteria and fungi. However, antibiotics are specific to bacteria only. So if you go on a broad spectrum antibiotic, that broad-spectrum antibiotic will be toxic to your microbiome of bacteria. Those bacteria will die off, and that provides an opportunity for room to grow for the potential pathogenic yeast or other types of fungi that can be in your body. Also, untreated diabetics. This is because diabetes results in excess sugar in the blood. However, it can also be excess sugar in saliva and sweat. And fungi love sugar, especially yeast. So they love to ferment excess sugar. So the more sugar, the better when it comes to promoting a fungal infection. Lastly, pregnant women and also breastfeeding women are at a high risk category for opportunistic fungal pathogens. So in this lecture, we're going to describe two of the common morphologies of pathogenic fungi. So the first morphology we're going to describe are the molds. So molds are filamentous fungi that reproduce by spores. Most of us have seen fungi growing at home on fruits or vegetables or on cheese or on spaghetti dinner in Tupperware containers left in the back of your refrigerator for weeks. Oh, come on, I know I'm not the only one that leaves Tupperware food in the refrigerator for weeks, and then eventually you open it up, and lo and behold, there's something fuzzy growing on that food. So it's possible to see molds with the naked eye, and most of us would describe the appearance of molds growing in the environment or on food objects as fuzzy, this fuzzy growth. And now we're going to learn the technical term, though, for fuzzy. The technical term refers to these branch-like filaments that the fungi produce, which are called hyphae. You can see that here on the screen. This is on the far left. We're looking at mold microscopically. So looking at mold microscopically, we see the components of the cell structures. So these filamentous cells are called hyphae. And the hyphae really have two functions for the mold. One function is to produce the spore structure called the sporangium. So the sporangium in this picture, this is actually bread mold, is what we're seeing here on the slide. And so we see these little pods that form at the tips of these hyphae branches. And we call these little pods, we call them sporangium. And so if you could look closely there, you'll notice that inside of the sporangium, we would find the spores. These are microscopic spores. So some of the hyphae, they terminate in these little buds called sporangium that are packed with spores. And then the other function of the hyphae would be feeding. So there's feeding hyphae. Sometimes feeding hyphae is referred to as vegetative hyphae. So this hyphae acts as an anchor. It looks very root-like. So root-like, and it anchors deep down into whatever that spore has landed on. So maybe a piece of bread or a piece of cheese or the skin or the lung tissue. Okay, so these fungi, they're looking for organic material to decompose. They are decomposers on organic material, living or non-living organic material. They are decomposers. So these feeding hyphae will act like deep roots. They'll go deep down into this organic material, and then it will release enzymes. And these enzymes will digest the organic material and basically liquefy it. Let's see that in time lapse, because this is pretty amazing in a time lapse to see these hyphae form and then liquefy their substrate. So if you follow with me the link at the bottom of this slide, if we click on it, let's take a look. So now we're going to watch in this time lapse these 3 beautiful strawberries become digested by a mold. So how does this happen? Well, there's microscopic spores in the air all the time. The spores land on a nutrient-rich surface like these strawberries and then begin to germinate into those branch-like fuzzy structures we call hyphae. Then they secrete enzymes that digest the strawberries extracellularly and liquefy them. And you begin to see hyphae form towards the top of the mold, and those would be our reproductive hyphae making the sporangious structures. And then the vegetative hyphae would be down below, liquefying the strawberries or feeding off of the strawberries. Okay, so we've got our feeding hyphae, and then we've got our reproductive hyphae. Usually the reproductive hyphae are at the top of the mold because they want to make sure that those spores do get released and spread either by wind or by water to a new nutrient source to feed and to continue to grow in its life cycle. On this slide, we see some additional microscopic pictures of some molds. On the left, we see a very famous mold, and this mold is the mold that produced the very first antibiotic. If you remember that story, the mold is called Penicillium. And Penicillium produces the chemical we call penicillin. So this is what Penicillium mold looks like under the microscope. And I just want to show you that there are different appearances to the sporangia portion of molds. So Penicillium sporangia under the microscope looks different than the pod-like sporangia that you see on the right. The right, this is actually a different mold called Aspergillus. And Aspergillus is an opportunistic fungal pathogen that can cause fungal pneumonia. Okay, and its sporangia looks more like a little pod, okay, pod shape or round, compared to the Penicillium sporangia, looks almost like a flower or like a little hand. And we see the spores here, almost like the fingers of this sporangia structure. All right, so there's some variety in how those sporangia appear under the microscope. On the topic of spores, let's pause a minute and talk about bacterial spores versus fungal spores. So this isn't the first time you've heard that word spore in our microbiology class. Recall, we've talked about bacterial spores before, and we even did the spore stain early in the semester, where you got a chance to see that certain species of bacteria, not all, okay, but certain species of bacteria are spore producing. And when we do the spore stain, as shown on the picture on the left, and you look at those spore producing bacteria under the microscope, what we observe are the vegetative cells in red. So these red bacilli that you see here, these would be what we call the vegetative cells. And then the green structures that we see either outside of the vegetative cells, okay, so we see some external spores, and those would be in green. And that's part of the spore stain, if you recall. Using the primary stain called malachite green, that drives the green color into the spore coating, which is why the spores appear green under the microscope. In this picture, we're also seeing some spores internally inside of the vegetative cells. And when they are inside of the vegetative cells, remember, we call them endospores. So what is the purpose of bacterial spores? Is this how the bacteria reproduces? No. Bacteria reproduce by binary fission. So spores are produced by certain species of bacteria as a way to protect the cell if the environmental conditions are adverse. Meaning, maybe they've run out of nutrients, maybe it's very hot, or it's there are chemicals in the environment. And so recall the spore actually is a dormant cell. So it's not a reproductive structure. It's a dormant cell. So certain vegetative cells will produce spores and release those spores sort of in a mechanism of what we call an SOS response, meaning that the environment is toxic to the vegetative cells, but if the vegetative cell goes into a hibernation form or a bacterial spore, then it can stay in that dormant stage for almost indefinitely, okay, in the environment, because bacterial spores are highly resistant to chemicals, to heat, to radiation. So this is a protective response that the bacteria does to go into sort of a dormant stage. Sorry, I meant to write bacterial spore here. So we would consider the bacterial spore to be a dormant stage in the life cycle of certain spore-producing bacteria. So it stays as a spore, and then it can, when the environmental conditions are favorable, regerminate into the vegetative cell. So that spore can regerminate into a single vegetative cell. And then that vegetative cell go and reform a spore. And so it goes through this process only in certain groups of bacteria as a dormant part of the life cycle. So very important to remember, we do not consider this reproduction. So it's not reproductive. Okay, so to contrast that with fungal spores, on the right, we are seeing, again, penicillium, sort of that flower-like appearance to its sporangia under the microscope and a whole bunch of spores being released. So in contrast, fungal spores are reproductive. So this is the reproductive structure for molds. Okay, so reproductive structure for molds. Okay, so in other words, if we have a fungal spore released by the sporangia of a mold, that fungal spore will go and it will germinate and form a new mold. This is hyphae here. Something like this. Okay, so it'll form these hyphae structures and it'll make new sporangia. We'll just make the pod shape to the sporangia. And then that sporangia will release new spores which create new molds. Okay, so that is part of the reproductive stage of the fungi. Now, fungal spores are also fairly resistant to heat, chemicals, and radiation, but not as much as bacterial spores. So bacterial spores are recognized to be more resistant to environmental conditions than fungal spores. But fungal spores can still persist in the environment, in soil, in moist environments, and then can regerminate into a whole new mold and produce lots of reproductive spores. Now we're going to talk about the second form of fungi, the second morphology, and that is a yeast. So yeast are unicellular. Single-celled, it's a unicellular fungus, and it reproduces by a process called budding. So if you see them under the microscope, yeast have this oval shape. So this is how they look under the microscope, oval shape, much larger than a bacterial cell. Okay, about 10 times the size of a bacterial cell under the microscope, with a classic oval shape. So when they reproduce, they form this little bud. So this little structure that's coming off the parent cell, so we would call this the parent cell. And then we would call this the asexual bud, because this is a process of asexual reproduction. In other words, the bud that's forming is identical genetically to the parent cell. There you go. On the right, let's watch this in time lapse. So we're seeing the budding yeast process in action. So watch some of these oval-shaped parent cells start to bud. Oh, I like this part too. So here we see the budding process in action. So it starts as a small projection from the cell that expands and grows in size until eventually the bud breaks off the parent cell. And now we have two identical cells. Ooh, here goes another one budding. So it buds off and breaks off, and there we go. That's the identical copy. Okay, more budding. Cool. So some pathogenic yeast have one little extra thing that they do here that I want to talk about when they form the buds. So some pathogenic yeast, when they bud, they actually form a branch in between the bud and the parental cell rather than breaking off. So in the previous slide, we saw breaking off, right? The bud would break off once it expands to equal size of the parent cell. It just breaks off. Here, we're seeing that the bud actually stays attached to the parent cell and forms a branch like filament between those structures. Now that's a lot like hyphae, okay? Looks a lot like hyphae, but we call it pseudo-hyphae. So all these little arrows, what they're pointing to this branch that's connecting, and we would call that pseudo-hyphae. Pseudo just means fake or not true. Hyphae. So the point is that it's similar to the hyphae that molds produce when they germinate from spores in the reproductive and the vegetative hyphae. But here we're talking about yeasts. We're talking about a parent yeast cell that forms a bud but then that bud elongates with this filament called the pseudohyphae. Why is this a structure that happens in pathogenic yeast? Well, let's take a look. It's actually a virulence trait of pathogenic yeast in the body that are able to do this. So I'm about to animate this and show you what happens here. So we're going to watch as an example of a pathogenic yeast who does this, by the way. It's actually an opportunistic pathogen, and this yeast is called Candida albicans. So Candida albicans is an opportunistic yeast, and what you're going to see in this animation is what happens if the yeast get engulfed by a macrophage. So what is a macrophage supposed to do? Oops. The macrophage is supposed to digest and eat pathogens, right? Phagocytosis. Well, that's all well and good unless microbes have a virulent strategy to resist phagocytosis. Take a look at what happens. So the macrophage engulfs it, and then the yeast forms a pseudohyphae and bursts the macrophage. Okay, so that brings us to our slide on fungal virulence factors. Pseudohyphae. This resists phagocytosis. It's a strategy of certain yeast that when they get engulfed by a phagocyte, they form a pseudohyphae like a big spear inside of the macrophage and breaks open that macrophage or that phagocyte. Other strategies, enzymes. So we watched the video of the strawberries becoming covered in the hyphae structures and being digested. So that's the strategy of feeding for fungal pathogens. Fungal toxins. A lot of them produce toxins. This can cause a number of different symptoms, including headaches, chills, severe nausea, vomiting, and hallucinations. Here's some examples. Aflatoxin is produced by Aspergillus molds. Aspergillus molds grow as black molds. So sometimes these show up in wallboards and contaminated foods. So black molds growing in usually moist environments like inside of bathrooms. And in severe cases, this can cause fungal pneumonia and has even been linked to lung cancer. Ergot. Ergot is a toxin that's produced by a species of mold called Claviceps. This is a fungal pathogen on plants, and if humans consume the food from these plants, they may experience hallucinations. And this is actually a natural source of the chemical LSD, okay? And then fungal spores that we mentioned earlier, resistant to heat, chemicals, and radiation. Now let's talk about how do we treat fungal infections. So this slide is titled Anti-fungal Medications, and we're looking at the structure of a fungal cell, okay? And we're looking at the targets used by antifungal medications. And the question I posed on this slide was, which antifungal medications shown on this slide are the most selectively toxic to fungal cells, not human cells? That's a really important part. That's a really important thing to understand about any medication that we use to treat patients. We talked about this concept when we talked about antibiotics, the concept of selective toxicity. And what that means is that you want the medication to be selectively killing the pathogen, but not harming the host, not harming the patient's cells. So what's easier to treat? A bacterial infection or a fungal infection? And why? Well, based on that principle of selective toxicity, bacterial cells are prokaryotes. Their cell structure is very different compared to eukaryotes, at least in several ways. When we studied antibiotics, we learned that antibiotics are specific to only bacterial cells. They only kill bacteria because antibiotics target certain cell structures that are unique to bacteria only. That is why antibiotics are not effective against other types of pathogens. So that means when we describe medication that treats fungal infections, please use the word antifungal rather than antibiotic, because antibiotic means medication that's designed to kill bacterial cells. Okay? Okay, so let's look at each of the types of antifungal medications on this slide and decide which is the most selectively toxic to the fungi and not the humans. So starting in the upper left, here we see antifungal meds that inhibit DNA and RNA synthesis of the fungus. Is that selectively toxic? No. We also, as eukaryotic cells, use the same or very similar enzymes for DNA and RNA synthesis. What about disrupting microtubule function? You might have to look up what that word means. It refers to specific proteins used by eukaryotic cells for cell division, like mitosis. That would not be selectively toxic. What about inhibiting mitochondrial function? Do your cells have mitochondria? Yes. So that would not be selectively toxic. What about disrupting cell membranes? We also have cell membranes. What about inhibiting ergosterol? Yes, this is selectively toxic. So we're highlighting the targets of antifungal medications that would be the most selectively toxic on the slide. So what's ergosterol? Well, ergosterol is actually a unique cholesterol molecule that's only found in fungal cells, in fungal membranes. Yay, we have something that's unique to the fungi that we don't have. So therefore, that would be a very good target that is more selectively toxic. How about over here? Inhibiting chitin synthesis. Chitin is the cell wall of fungi. So all fungi have a cell wall made out of chitin. Do we have a cell wall? No, animal cells do not. So chitin is the component of the cell wall, fungal cell walls. And then ergosterol, this is a unique cholesterol found in fungal cell membranes. What about synthesis of beta glucans? Well, that's part of their cell wall. We don't have those. So this strategy would also be selectively toxic. So that means the majority of antifungal medications that are prescribed fall into one of these three categories. That would be the first choice for treatment, would be to use a antifungal medication that is selectively toxic to the fungi but not the humans. Next, we're going to talk about some examples of pathogenic fungal mycoses. So remember, the most common fungal mycoses are cutaneous or infections of the skin, and the second most common are respiratory mycoses. So first, cutaneous. So this is caused by a group of molds we call dermatophytes. They grow on skin, hair, and nails, and some examples are shown here. So we see this man here with several of these red ring spots of inflammation on the skin, and this is referred to as ringworm, or sometimes referred to as tinnias. So ringworm is a little bit of a misnomer. There's not a worm that causes this rash. Please don't confuse that with infections caused by actual worms, which we will be getting to in the third video in eukaryotic pathogens this week. So it's not caused by a worm. It does have a ring shape to it though. Okay, so they can be very large ring shapes on the skin, and these spores land on the skin and they germinate into molds that cause a localized inflammatory response on the skin. On this side of the screen with these people's feet and in between the toes, this is referred to as athlete's foot, commonly. Well, athletes are more likely to have excess moisture on their bodies from sweating. This is why it becomes really important to wash your clothing after you work out. Maybe when you come home from the gym, good idea to shower and change clothes. That's because sweaty clothing can accumulate moisture and provide perfect growth conditions for fungi that are just in your natural environment. Also, be very careful about public showers because cutaneous dermatophyte fungi live in moist areas like in shower stalls. If the public showers are not bleached regularly, it's possible to contract a fungal foot infection from using a public shower. That's why it's always a good idea to wear flip-flops in the shower if you're going into a public shower. Let's talk about Candida albicans. So this is the opportunistic yeast that lives in our bodies. And so what usually happens is one of those risk factors to initiate this opportunistic pathogen to cause actually a disease. So oral infections are called thrush. This is shown by thick white deposits on the tongue, as you can see in this picture. Also cause of vaginal yeast infections, itchiness, soreness, sometimes a discharge. Skin infections, so commonly like diaper rash. Usually these have little red bumps, raised bumps, red and inflamed. In severe cases, Candida sepsis. This is a very severe case of a yeast infection because the Candida will overgrow in the large intestine, enter the blood, and then colonize multiple organs, sometimes even the brain. And this can be very problematic to the person where they can have long-term Candida sepsis and severe fatigue and brain fog caused by this yeast infection. So go back to the slide about the risk factors for developing fungal disease and why Candida albicans acts as an opportunistic. Lastly, we're going to talk about the respiratory pathogens, so respiratory mycoses. So something that respiratory mycoses, at least the two we're going to describe here in our lecture, something they have in common is they're both dimorphic fungi. So what does that word mean? Well, the word di means two, and the word morph means shape. So this means a fungus that has two shapes. So what are the two shapes that we've learned about in this lecture? The mold shape, spores that germinate into hyphae structures, or yeast, oval-shaped single cells that divide by budding. Okay, so it's possible for dimorphic fungi to exist, and this is usually temperature dependent. So inside the body, it acts as a yeast or grows as a yeast and looks like a yeast. So 37 degrees Celsius, this would be body temperature. 25 degrees, this would be the sort of the room temperature, environmental temperature. It acts as a mold. So it can act as a mold in the environment. And that's because that would be advantageous to produce spores in the environment that can reside in soil for long term. And then if somebody inhales those spores, maybe there's a lot of wind, like when we get those Santa Ana winds that start blowing here in Southern California, and you get lots and lots of fungal spores in the air. Now, most of the time, that's not a problem for you, unless you fall into one of those categories from the first slide where we talked about risk factors for opportunistic fungal pathogens. So any of those risk factors that might contribute to why you might develop a respiratory infection caused by a respiratory fungal pathogen. So yeast are less environmentally resistant than fungal spores. So yeast don't form spores. And so inside the body, they are better protected as or are able to survive as yeast, but outside the body, they would want to be protected as fungal spores. So the two examples of respiratory mycoses we're going to talk about would be blastomycoses and coccidiomycoses. So blastomycosis here, this is showing us again, it's dimorphic. Now this is on the map from the CDC is something that we see in more of the Midwest and the eastern United States. So you can see from this picture that we have the portal of entry being the nose or potentially the mouth. So breathing it in gets into the respiratory airways as spores and then grows into yeast. But outside of the body in the environmental form, it's in the mold structures that produce those sporangia. It's also possible to get a skin infection caused by this particular fungus, the blastomyces. So it's possible to get that as well, besides the fungal pneumonia. Now, just again, to reiterate, this is not a very common disease, and it's really associated with people who have underlying health conditions or are very young or very old. And it's like less than 1% of people who come down with this type of fungal pathogen actually develop fungal pneumonia that's severe. So the version that we have in the western United States, specifically the Southwest, is called valley fever. So that's what we have here in California. It's caused by a different pathogen, a fungal pathogen, and it causes coccidioidomycosis. And here's a little flyer about it. It talks about how, this can even transmit to wild animals, and this is going to occur if there's a lot of wind that's moving soil around, and there's soil in the air. So very few people actually develop severe infections from this disease, but it is an example of respiratory mycoses. So that's what we're going to end with today, and we'll move on to more eukaryotic pathogens in the next two videos.